Matsu Inc. is a company founded by one of Japanese famous deep learning laboratories, Matsu Labo, where I am working to apply deep learning research to various industries. In addition, I enjoy competing in data science competitions. Have you heard of Kaggle? Kaggle is the world's largest competition platform, where competitions on various tasks are held regularly, and data scientists from all over the world are competing day and night to solve these challenges. I have done well in several competitions on Kaggle and currently hold the title of Competition Master. Based on my research internship and Kaggle experience, I would like to talk about how data science is being used and what is interesting about it. First, I would like to divide life science research into 4 stages and explain them. Research in life science can be divided into 4 levels: molecular, cellular, tissue, and organisms. Molecular level research examines how molecules such as DNA and proteins interact with each other and what physical properties they have, such as degradation rates. Molecular level research examines how molecules, the next step is to examine how the molecules actually work in cells and how they affect the cells. It is common to see interactions at the molecular level, but not at the cellular level. This is because the inside of a cell is so complex that it is difficult to observe what is really going on. The third is the tissue level. Several types of cells communicate with each other to make up a tissue. It is important to study how each type of cells in the tissue works and affects the tissue. This is because studies at tissue level are more closely related to the living organisms and therefore more directly related to disease research and drug research. The last one is organism level. The phenotype at organism level is very complex because it is a combination of all the previous factors. In addition, it is important to find phenotypes that have not been identified before. Today, I will show you a few studies where data science is used at each level. The first two are from a Kaggle competition, and the other two are close to what I usually do in my research. Okay, let's begin with molecular level. First example I will show is mRNA vaccine. So you may already know, I would like to start by briefly introducing mRNA. Most cells in your body have DNA, which is storage of information. The mRNA is a copy of a specific region of DNA and is very fragile. The mRNA is then used to make the proteins that do the actual work. Basically, mRNA is produced when it is needed. It is degraded when the protein is made and is no longer needed. In this way, the amount of protein is regulated. Therefore, it is convenient for the organism that mRNA is easily degraded. In the mRNA vaccines, mRNA, which makes a part of one of the virus proteins, is injected to the body. The mRNA then creates the viral proteins in the body. The virus protein is then used to informs the immune system about the virus. This way, when the virus really enters the body, the immune system can work immediately. Here is a simple explanation of how mRNA vaccines work. The problem with mRNA vaccine is that mRNA is a very fragile substance. So even if it is injected into the body as a vaccine, there is a possibility that it will be broken down quickly. To prevent this, how to maintain the stability of mRNA is a very important topic. And stability of mRNA depends on the secondary structure of mRNA. The mRNA is made up of A, U, G, and C. A and U and G and C form a pair like this. The paired regions are less prone to degradation, but unpaired single-strand regions are more prone to degradation. And sequence of mRNA plays a major role in how the secondary structure of mRNA is formed. Predicting which sequences are stable and which are fragile will be important for the stable storage of mRNA vaccine. A competition was held on cargo last fall to create a model to predict this. The competition was to predict reactivity and degradability of each base based on sequence and secondary structure data. About 1,600 teams participated, and the top teams actually contributed to optimizing their models even after the competition was over. About sequence data used in the competition, the organizations that held the competition originally used a game called Etana to explore the structure of stable RNA in a game-like format. The game uses an algorithm called Tree developed by Etana to predict the stability of RNA sequence created by the game participants, and it gives them a score. It's a game like this. So Etana had a lot of RNA sequence information. Then, how were the labels created to predict the stability of the sequence? It can be determined experimentally using biochemical methods as shown in figure here. And this is actual target. However, this experiment is very expensive and noisy, so it would be very nice if we could make a machine learning model to predict it. I won't go into the actual solution, but various methods of deep learning are used and the results are very interesting. The top teams have shared their solutions and also shared some very helpful baseline code. So please take a look. Okay. Next, let's talk about the cellular level. Today, I would like to introduce cell morphology profiling. This is a study of cell morphology by staining some of the cell parts organella with different colors and imaging them. For example, nuclei are blue, mitochondria are red. It's like this. The images produced by this imaging process contain a variety of information. Staining intensity, texture patterns, size, shape of structure, information across channels, relationship between cells. In this experiment, we will examine the morphology of cells when they are exposed to inhibitor or stimuli. To examine the image of the cells, we will use deep learning method. that have been developed recently, such as convolutional neural networks. In the past, researchers would look at an image and determine, for example, that one organelle is clustered next to another. However, when deep learning models are trained well, it is now possible to extract information that humans cannot find. Another reason why we use the deep learning method for this research is that the number of images is very large. We use a plate full of holes like this to study a very large number of conditions. It is difficult for researchers to comprehensively analyze this large amount of data. On the other hand, with such a large amount of data, deep learning models are able to find better features. There is a company called Recursion that makes this kind of data available to the public. This company has released 4 datasets so far. As you can see here, it is very large dataset. In this presentation, I will introduce this dataset. A Kaggle competition was held using this dataset. I won't present here, but there are 2 datasets about COVID-19. The data size is very large, but it is very attractive dataset, so please try to analyze it. Okay, about this dataset. An siRNA is an RNA. An siRNA is an RNA that, when applied to a cell, disables the function of a specific gene. There are more than 1,100 siRNAs used in this dataset. The images of the cells treated with each siRNA are given. They are then used to train models and predict the unlabeled cells. In the end, the top team was able to create a model with an accuracy of over 99%. If the model looks at the image, it can tell which of the 1,100 siRNAs the cells was treated with. This is an amazing result. As I mentioned earlier, this dataset is open and available to anyone. In addition, the information of each siRNA is given after the competition was over. So we can see which molecules the siRNAs actually inhibited. So what interesting research can we do with this data? I haven't tried it, but I have an idea. First, we prepare the same cells in our own laboratory, and then we apply the procedure of interest that we are currently studying to the cell. Then we stain the cell in the same way as in the dataset. The resulting images are then classified by a model trained on a previously published dataset. Then we can see which morphologies of our cells are similar to those of the siRNA-treated cells. As a result, we should be able to predict the molecular mechanisms of what we are studying. Of course, there are a few issues that needed to be resolved, but if this works, I think it will be a very powerful tool. Okay, now let's move on to the tissue level. I will start by explaining RNA sequencing, which is often used in life science research. RNA sequencing allows us to look at all the RNA expression levels in a sample of interest, such as organ or cells, by using next-generation sequencing. By comprehensively looking at the mRNA expressed in the sample, we can see what kind of condition the cell is in, or the organ is in. This technology has been further developed in recent years. We can now analyze RNA expression at the level of single cell rather than tissue. By looking at all the mRNA expressed in each cell, we can now see what types of cells makes up the tissue. We take the tissue and separate the cell one by one. We then look at the expression of RNA in each individual cell. The sensitivity of the machine has improved so that we can now detect RNA expression from such small samples. Okay, I will now show an example of how single-cell RNA sequencing was used in one region of the brain. This area of the brain is known as the area that controls circadian rhythms. However, we did not know what types of cells are in this region, how they are distributed, and how they work. So the authors decided to investigate the cells in this region in detail by performing single-cell RNA sequencing. As I mentioned earlier, the authors took a region of interest and dissected it into single cells and look at the level of expression in each cell. Each cell's gene information. This gives us gene expression data at single-cell level like this. We can then use this data to find cell types by clustering such as PCA, t-SNE, and DP-scan. In this paper, cells were clustered by applying the graph-based smart local moving algorithms, and t-SNE was used as a dimensional reduction method to visualize the clusters. In this way, we are able to find classes of a few cells like this. In addition, genes that are uniquely expressed in each group can be used as marker gene for other studies. The final step in this paper is to make tissue slices stained for marker genes. They slice the mouse brain from front to back and stain each slice with marker gene. For example, on the left, the marker genes ABP, CCK, FOS are stained in different colors. Then they mapped out in 3D where each type of cell is distributed in the region of interest. These three-dimensional representations of the location of neurons may be useful in future studies of neural networks. Finally, I'll briefly talk about organism levels. In this level, what I found interesting this day is the use of video to analyze the behavior of mice. In this paper submitted to the journal Science in 2020, they recorded the behavior of mice from above. They then used UNet to perform segmentation and analyze the mouse behavior. Based on the result of the segmentation, the authors have defined two values, translation and total movement. By using these values and clustering them, They have found 3 new patterns of behavior: locomotor activity, non-locomotor, and rest. The authors then looked at the videos and labeled these 3 patterns. Then, using random forest, they created a model that predicts these 3 patterns from translation and total movements. This is a pipeline used in this paper to determine the behavioral patterns of mice. Later in this paper, they investigated the relationship between neural activity and these behavioral patterns. In this paper, they are not doing single RNA sequencing, but as I mentioned earlier, they are looking at when certain regions of the brain are made dysfunctional, how this pattern changes in mice compared to normal mice. Without this pipeline, they would have to check and label each video with their own eyes. That's a lot of work and it's not objective. It is possible that they would not have found these patterns of behavior in the first place. In my opinion, this is a good example of the effective use of this science. I won't go into details, so please have a look at the paper if you're interested. A tool that can be used by non-computer science research to recognize behavioral patterns in mice have recently published. The tool name is Deep LabCat. It has a GUI and easy to use. Accurate recognition with less labeling using transfer learning. It can be used for various animals, mouse, or flies, or other animals. I used it before and I feel it was very easy to use and recognize the mouse correctly. I believe that by using these tools, we can find new patterns of behavior phenotypes in animals. Okay, conclusion. This science can be used to find phenomena that could not be analyzed before. And there is a lot of biological data available, so anyone can use it for analysis. If you feel any interest in life science research and would like to apply this science technology to it, I'm very glad. Thank you. Any questions? How can we start in this field without any privacy?
Hello, sir.
Okay.
Sir, I wanted to know if we could use the same technology in uh, keeping a track of how the human race is evolving, and we could maybe interfere with the evolution of the species as a whole.
If we, sorry, I'm not good at English, so can you write the chat box? Okay. Anyway, first I answer the question, how can we start in this field without any previous experience? Uh, anyway, you try to analyze the open data like this, like this, or you can also available this data. So try it. Okay, where's good place to post the project we do with it? Oh, so the this data set is maybe not posted to bioarchive. Sorry, I forgot, but any other data set of cell morphology is posted to nature methods. So it's very high impact factor. Can we use the same technology to trace? Ah, I see. Sorry, I'm not familiar with the evaluation now field. So I don't know that we can use the same technology to this kind of field. I will share the slide, the material later. So if you have question about this slide, please send me message by Twitter or LinkedIn. This is my account. Oh, I missed the message. Oh, can we use the same technology? You can share and solve data science related problems on Kaggle. Evolution. When did you start all this? Okay. The question is, when did you start all this? How did you develop interest in this field? I started learning this data science from Kaggle. At first, I tried Kaggle competition. Then I'm very fascinated in this field. Thank you for the question. Recently, I was working on a project to track. Okay, the question is, recently I was working on a project to track migration movement of 3 girls. I want to know where I can find more such research-based datasets. Uh, I you can find datasets is paper with code. Paper with code is a website about machine learning or deep learning research, and this site also shares the dataset information. So please check this website. タイトルここですか Good sensation. Can we use a classing for these types? Can you type the URL? Oh, okay. Can we use crossing eyes for these? I have GitHub account, but I'm not upload useful code. Okay, my GitHub account is okay. I shared GitHub account. Next, if I got good praise on Kaggle, I will share my code. Seeing number of participants who have turned up the session. How is Japan for research work? It is very good place. Uh, the level of research in biology is high, but I don't know about machine learning. Kyoto give. Yeah, Kyoto University I belong. Many Nobel Prize holder.好きなアニメ。アニメ。I think Ghibli is very good animation. Is mathematics and computer science background enough to get certificates? Mathematics. Uh, is mathematics and computer science background enough to get started with data science in the field of biology? Yes. Uh, a computer scientist would be able to understand biology very quickly, I think. Yeah, you can also always message me on Twitter. I'm the particular student. Uh, the question, I'm biotechnology student. I have interest in this field. Where should I start in this field as fresher, as I don't know have enough knowledge? Try to use the data anyway for this field. Try to analyze open data. Let's this show it here. How to not overfit public LP. Uh, Trust your local cross-validation. Um, I recommend this dataset because this dataset is very interesting and the recursion published the one of their analysis about COVID-19 data. And yeah, it is true that this dataset is very large, but the low dimensional embeddings, they also share more small data about this. So try it. You can analyze this small data by Google Colab. So if you are interested, please try this dataset. The question is easy for a non-medical background person apply data science in field of biology? I think it's easy, but I think it would be more interesting if you have some knowledge of biology. So please study it. The question is, can you recommend some books for data science in field of biology? Uh, Sorry, I'm not familiar with English books, but I think it's better to read papers in this field. You can access to bioarchive, so try to read papers. What are the specifications of your PC or laptop? Uh, I always use two GPUs. Recently released RTX 3060, and CPU is I like AMD CPUs. But you can start not so expensive resource. You can start Google Crabble.
Okay, thank you, sir. I would like to ask if anyone has any else question for now. Okay, since we don't have any more questions, we would like to thank sir for giving his time. And we will be also uploading the session on YouTube so that maximum of us could benefit from it.
Okay, thank you.
Thank you so much.
Thank you, sir. Let's conclude this session because of some time constraint.
Okay, thank you.
Yeah, thank you, sir. It was really a wonderful session. Yeah. And we hope to see you soon in our future events too, sir.
Thank you.